Hello, everyone, and welcome to another exciting episode of the Biologic Podcast. Today's show is episode 115, and we're going to be starting a whole new series in this multi-part super series on the protostome animals. Our first series was about the mollusks, with episodes covering each major mollusk clade. The next series introduced the arthropods, beginning with the most basal of the major clades, the arachnids, and their chelicerate relatives. That was concluded in the previous episode, and it was super interesting. Spiders and scorpions are just awesome, and that whole series was really cool. Continuing in this arthropod multi-series, our next 6 episodes are going to be all about the myriapods. These include the centipedes of the class Chilopoda, the millipedes of the class Diplopoda, and their less well-known but equally creepy-crawly relatives in the classes Pauropoda and Symphyla. We'll take a look at each of these classes in the following episodes, but today we're just going to set the stage by exploring their early evolution. This is so that we can learn how and when they emerged into the world, what ecological niches they evolved to fill, what the world was like at the time they emerged, and what demands and pressures were placed upon them, and how they've diversified into the modern myriapod menagerie that we know and love today. To briefly summarize, these are the Symphyla and the Pauropoda, which are both small, usually pale or translucent, millipede-like creatures that live in the soil in habitats across the planet. The millipedes, which are soil-dwelling herbivorous consumers of plant detritus that can be found in forests and vegetated areas across the world, and the centipedes, which are the only carnivorous predatory myriapods. If you want to hear more about the myriapods as a clade, or if you want to hear about one or more of the classes in greater detail, you should go back and listen to episode 88, Arthropoda Part 2, the Myriapods. This episode is a great primer on the clade, as it introduces the phylogenetic relationships of the 4 modern classes in a nice, concise way. Now, before we get going with all of the evolutionary history, let me start by explaining a few important details and clarifications about today's topic. Same as we saw with the mollusks, the early fossil record of the myriapods is pretty meager, and it does not give us an intrinsically accurate, high-resolution understanding of the animals as they lived over 500 million years ago. Mollusks are extra tricky because so much of their body is soft tissue, and it rapidly dissolves after death, so those parts rarely get fossilized. Usually, the only thing that does is the hard, mineralized shell. It's the only part of them that remains. And in many cases, they don't even have a shell. Now, the myriapods have a chitinous exoskeleton across their entire body, so they don't have the really soft tissue of an octopus or a squid or a slug. But this exoskeleton isn't very hard either. It's certainly not as hard as the mineralized shell of a snail or a nautilus, and it's not as hard as most vertebrate bones either. So myriapod exoskeletons are more likely to fossilize than the soft tissue of an octopus or a squid or a slug, but they're not as likely to fossilize as vertebrate bones. This means that the fossil record, especially their early fossil record, is relatively sparse and cryptic. To make matters worse, modern myriapods tend to concentrate in heavily vegetated, often deeply inland areas. Think jungles and temperate forests. These habitats have environmental and biological variables that are much more likely to quickly consume, degrade, and dissolve a corpse right down to the bone than they are to fossilize it over a prolonged period of time. For example, these habitats are notorious for having acidic soil, and this helps to accelerate the breakdown of even hard tissues like bones. As a further complication, of all the macroscopic terrestrial animals in the world, the myriapods are among the most mysterious and unfamiliar. They're very discreet as they live in soils and leaf litter and plant detritus, so they often go unseen. They're also relatively understudied. Genetic research has only constructed high-resolution molecular phylogenies in the last decade or so, and it's displaced a lot of the earlier morphological research. It turns out that those earlier morphological studies were not really using a reliable method to establish the taxonomic relationships of the various myriapod classes. When more sophisticated measurement techniques led to more sophisticated and more accurate hypotheses, the older hypotheses built on those outdated morphological studies were basically thrown out. For example, it was once thought that the myriapods were paraphyletic with insects. This is to say that the ancestral myriapod was actually a hexapod, which would mean that all myriapods are technically hexapods, just as all hexapods are technically crustaceans, but just radically different in a few ways and given their own special name and clade to distinguish them. But more recently, it's been established that the Myriapoda are monophyletic, and they're not descended from insects at all. In fact, all the myriapods, all 4 classes, are the descendants of a common ancestor that is not a hexapod, but was instead related to the common ancestor of the crustaceans. So if we take a step back and look at the larger arthropod phylum, within this whole arrangement, the Chelicerata, which includes the arachnids and the sea spiders, is the most basal group. It's the most basal offshoot of the stem lineage. It represents the first major surviving arthropod divergence, and it produced all of the scorpions and spiders and everything else. The stem group, or the original line from which the Chelicerata diverged, became the Mandibulata. It was basically the sister lineage to the spiders and scorpions. And it is this Mandibulata lineage from which the myriapods would soon diverge. After the ancestral myriapods split off and diverged on their own, the remaining Mandibulata would go on to produce the Pancrustacea. This extremely large clade would produce all the classic crustaceans, and from within them would emerge the hexapods, and within this hexapod lineage would come the insects. So that's basically the placement of Myriapoda within Arthropoda. They're kind of basal, not as much as the spiders and scorpions, but they're more near the middle of the family tree. They're younger than the spiders and scorpions, but they're older than the crabs and the lobsters, and of course the insects that came from them. Although recent studies have suggested that they're more closely related to crustaceans than they are to arachnids, so this kind of gives you an idea of where that branch is positioned on the tree. Is it closer to the first branch or closer to the later branch? It apparently turns out it's closer to the later branch. Now, figuring all of this out was really a challenge. It took quite a while to figure out where to place the myriapods in relation to the insects and the arachnids and the crustaceans. Morphological studies were inconclusive, or worse, counterproductive, and it really took modern molecular research to try to clear things up. But for years after molecular research was developed, there's still remained some debate and some mystery about how each of the 4 classes of myriapod are related to each other within the clade, even after analysis with modern instruments and genetic sequencing. When I was doing research for this episode, I noticed that more recent genetic studies conducted in, say, the late 2010s were building revised, updated, and different phylogenies than genetic studies from just 4 or 5 years previously, from the mid-2000s. Things are apparently changing pretty quickly in this field of research, as the latest techniques are being applied and the data interpreted. For example, 2 major phylogenetic studies performed within the last decade came to conflicting conclusions. A paper from 2018 suggested that Parapoda and the millipedes were sister taxa in a clade called Dignatha, while a paper from 2020 suggested that the Parapoda were actually more closely related to the Symphyla, with the centipedes and the millipedes forming their own sister taxa. A paper published in 2021 reported what was arguably the largest myriapod genetic analysis ever conducted, including all of the species used in the previous 2 studies. This 2021 study produced some of the most robust data ever constructed on myriapod phylogenetics. The paper argues that an older phylogenetic conception called Dignatha, which saw the millipedes or the diplopods closely related to the parapods, is definitively false and can be rejected. In its place, they confirm the general findings of the 2020 paper, where the symphylan and Parapoda are closely related sister taxa, and the millipedes and the centipedes form a second closely related sister taxa. The 2020 and 2021 papers differed slightly in arrangement of species within the Chilopoda and Diplopoda, but otherwise they were quite similar, and they provide strong evidence for the proposed phylogenetic arrangement. In addition, both the Diplopoda and the Chilopoda would become extremely diverse compared to their smaller, simpler Symphyla and Parapoda cousins. For the rest of this series on the myriapods, we're going to go in this order. You know how I like to arrange these things. I like to have the oldest clades first, and as we go through the episodes, we explore increasingly derived clades until we get to the most recent ones, the youngest lineages. Alright, so that was maybe more than a few clarifications and tidbits of background info. But at last, now we can finally go on and tell the story of the evolutionary history of the myriapods. Let's go far back, long before the ancestral myriapods first appeared. Let's start way back when the ancestral arthropods emerged. The earliest arthropod fossils are over 500 million years old, representing a lineage that emerged during, but more likely quite some time before, the Cambrian explosion, when complex life was still limited to the oceans. This ground-dwelling aquatic creature, this common ancestor to the arthropods, was a small, oblong organism with a body demarcated by clear segmentation. At the front, it had antennae and eyes, organs that it used to sense the outer world. Along the underside of the head, they possessed a circular mouth lined with small teeth. It was likely an active predator that hunted through the sediment. It's possible that they may have preyed upon primitive mollusks, among other things. If you recall from the previous series, around this time, 500 million years ago give or take, the ancestral mollusks were little more than slug-like creatures creeping on the seafloor. These animals hadn't even evolved to swim yet. Even mineralized shells were a work in progress, as an evolutionary response to predators swimming above them, or crawling on the seafloor beside them and rearing their head over them and striking down at their backs. Speaking of ancient mollusks and ancient arthropods, it's also really interesting to note the similarities of these two ancestral forms. They were both bilaterally symmetrical invertebrates, with a forward-facing head equipped with eyes and sensory structures, and a downward-facing mouth. Where the mollusk ancestors evolved shells that they could retract their soft tissues into for protection, the arthropod ancestors evolved harder exoskeletons across their entire body. This stiffening of the tissue was necessarily associated with the evolution of segmentation, which provided joints in the armor, so to speak, to allow the arthropods to be flexible and move around, to bend their legs and stuff like that. This trait, this segmentation, would become extremely important for the arthropods. First of all, segmentation was a process exploited by the Hox genes, which are a set of genes even older than the arthropods and the mollusks. These are a consolidated toolkit of genes that are used to organize the structure of the body and its appendages, particularly body parts that are similar or repetitive. Think like all of an organism's limbs, like its arms and legs, which are all pretty similar, or your fingers and toes. By running the same set of genetic programs over and over again with minor tweaks and modifications here and there, the genes can very efficiently produce the same proteins that produce the same body structures over and over again. This gives you a set of similar limbs, like all the legs that you see in spiders and crabs, and of course, all the legs in the myriapods. These genetic programs can be modified or tweaked slightly to produce different types of appendages, such as antennae, or mandibles, or claws. These Hox genes are so important that they've been preserved all this time. The Hox genes regulate the body plans of flies, lizards, lobsters, birds, centipedes, and humans. In fact, our arms and legs and our fingers and toes, they're all organized with Hox genes, just with small modifications that make our fingers long and tactile compared to our toes, and our legs thick and strong compared to our arms. Coming back around to the arthropods. In the more basal chelicerate lineage, body segmentation produced forms like the spider with its oval-shaped body segments, 8 long legs and its bulbous pedipalps, or the scorpion with its curling series of nested plates tapering into a tail that ends in a fierce and venomous stinger, and the foremost 2 major appendages which have been turned into giant pincers. In the crustaceans, a variety of body plans emerged, including the curling prawn form, the lobster form with its characteristic pincers, and the crab, whose body form has independently evolved so many times that the phenomenon has a name: carcinization. In the hexapods, the body generally has 3 segments. There's the head, the thorax, and the abdomen, with 6 legs and antennae. In the context of myriapod evolution, the myriapod strategy appears to have been spamming the repeat button on these Hox genes. They evolved to run these Hox genetic programs over and over and over again. A typical myriapod body segment included some thicker dorsal plating on top, a few small respiratory and metabolic organs embedded inside, and a pair of limbs, all supplied by primitive vasculature that passes through each segment, funneling oxygen and nutrient-laden hemolymph down the length of the body. The myriapods evolved to have many such body segments. They've evolved a process called anastomosis, wherein they can add new abdominal body segments during each molt, repeating this process over and over again to create a massively elongated, mature body form. These traits can be seen in primitive mandibulata fossils, in the species that lived after the arachnids diverged, but before the modern myriapods themselves had emerged. There are several mandibulata fossil specimens that are understood to be an early ancestral arthropod. For example, Sprigina is a small, primitive-looking, bilaterally symmetrical creature with a simple half-circle head, followed by rows of plates in a tapering tail section. It very well looks like it could be the ancestor of trilobites, and of myriapods, and of all the other arthropods for that matter, but it lacks certain traits, like jointed legs, that would definitively tie it in as an arthropod ancestor. Now consider those famous trilobites. These are definitely early primitive arthropods, and together with the chelicerates, they form a clade called Arachnomorpha. The trilobites are small creatures with hard, multiplated exoskeletons that fossilize pretty easily. They've been preserved in exquisite detail from sites all across the planet, and from this very comprehensive fossil record, we've learned that they emerged about 521 million years ago, demonstrating a very high level of pre-existing diversity, and they continued on, existing in some form or fashion, for another 270 million years. Now, the trilobites are an interesting example, because they're widely seen as representing the archetypical ancestral marine arthropod, even though they themselves are actually an offshoot branch and not the actual ancestors of the arthropods. It's more likely that they have a common ancestor with the arachnids. As such, the trilobites represent a very close cousin to the basal chelicerates, and a somewhat close cousin to the mandibulata, or the mandibulate arthropods. The name of this clade, mandibulata, comes from the presence of jaw structures, or mandibles, distinct from the chelicera and pedipalps of the arachnids. Once we get into the mandibulata proper and start examining the fossil record, we'll see many early species that are armor-plated, with a wide head and a tapering body, and a squirming mass of legs running along the belly. They resemble both their older cousin trilobites and, in a curious way, the myriapods, who would soon come after them. Consider the Euthycarcinoidea, which is suspected of being an ancestor to the myriapods. This creature emerged in the oceans in the early Cambrian, and they survived for the entire Paleozoic era. Within 100 million years of their emergence in their saltwater habitats, they had moved inland, perhaps seeking more abundant food in shallower, sunnier coastal waters. They evolved, adapting to brackish habitats along the coast, like estuaries or mangroves, while others went further and adapted to freshwater habitats like rivers and lakes. It's believed that they fed on thick bacterial mats and molds and other slimy biochemical-laden goop on the bed of a lake or a river or along the coastal shelf. The core of the Euthycarcinoidean body was a long, segmented cylinder, with each segment sprouting a pair of legs. A hood or mantle or some kind of broad segmented plating lined the back, extending outwards above the segments with legs. A few more segments, equipped with neither legs nor armor plating, extended posteriorly to form a tail. Standing back, you can almost see the evolution in motion. The Euthycarcinoidea had a common ancestor with trilobites, no doubt where they both got their dorsal armor plating from. But the Euthycarcinoidea are also narrowing and elongating their bodies, taking on a much more myriapod-like form. Their wide-set eyes and antennae already appear to be well-formed 500 million years ago, and they really haven't changed that much since. Another ancient extinct clade is the Fushenhuida, which existed from at least 520 to about 509 million years ago. Much like the trilobites, the Fushenhuida were benthic creatures. They dwelled on the seafloor, where they crawled along in search of food. They too had well-developed eyes and antennae, which they may have used to detect prey to hunt or dead bodies to consume. There's evidence suggesting these creatures were carnivores. Found exclusively in Yunnan province in China, all of the fossilized individuals possess a hard mouthpart called a hyposome, and a number of sharp, specialized, shell-breaking limbs near the mouth called gnathobases. These traits, this hyposome and the gnathobases, imply that at least some of the Fushenhuida genera, like Alacaris and Chengjiangocaris, hunted and fed on animals with hard shells. At the time, these would have included primitive mollusks like the protogastropod Halcyon-elliforms, and primitive bivalves like the Pogitaia. Most relevant to the myriapods, the Fushenhuida had an elongated body composed of many repeating segments. Like the trilobites, the early Fushenhuida had their massive legs concentrated near the thorax, towards the front of the body. But as longer bodies were evolved, so too did the number and distribution of the legs. Later species to emerge, like those in the Guangwicaris genus, showed evidence of elongation. The abdomen was significantly longer and more consistent in size, instead of tapering towards the end. The massive legs, once concentrated under the thorax, were now protruding from more and more segments running farther down the body. Aquilonifer spinosus is a more recent specimen, from the mid-Silurian about 430 million years ago, and it shows a remarkably more myriapod-like body form. It still has two large tentacle-like antennae, but the body is no longer tapering at all. It's pretty much entirely cylindrical, with legs protruding outwards from each side, equidistantly, down the entire length of the body. Despite how it may seem, the Aquilonifer spinosus is not an ancestor of the modern myriapods. It's more like a now-extinct cousin of the myriapod common ancestor. These ancestral myriapods, the common ancestor and its immediate predecessors, actually existed as far back as the late Ordovician, about 450 million years ago. But all of the biodiversity that was cultivated in these first 20 million years or so of their existence has been cast into extinction. It wasn't until about 430 to 428 million years or so when the first millipedes started to appear, at least as is documented by the fossil record. This is to say, the first millipede fossils are dated to around 430 to 428 million years ago. Centipede fossils appeared around the same time, a little bit later, but around the same time, although the centipedes seem to be pretty rare up until the Mesozoic, when they begin to appear in the fossil record with much greater abundance and diversity. Curiously, the Symphyla and the Parapoda are both poorly represented in the fossil record, and what few fossils there are tend to be mostly from Cenozoic era amber. In other words, If we just go by the fossil record, it would appear that the Symphyla are about 100 million years old, and the Pauropoda are a mere 40 to 50 million years old. This is almost certainly not the accurate ages of these lineages, for two big reasons. First of all, the bodies of Symphyla and Pauropoda are more likely to be destroyed by predators, scavengers, fungi, or natural forces than they are to be fossilized. And when they do form fossils, they're extremely easy to erode or destroy. In the cases where the fossils do form, and they aren't destroyed over millions of years, they're very tiny and very hard to see, and almost certainly have eroded at least partially to the point that no useful information can be gained. And this is, of course, if they're even recognized in the first place. So it's really not surprising at all that their fossil record is very, very sparse. And the second thing is that there's very strong genetic and molecular evidence suggesting that the Symphyla and the Pauropoda are actually from a lineage that's older than the centipedes and the millipedes. They represent an older, obscure cousin lineage that branched off at the root of the myriapod clade and has spent the last 500 million years meandering around in the soil, blind, evolving very slowly, and diverging very infrequently. In episode 88, I briefly explored the 4 major clades of living myriapods. I also mentioned a 5th clade that had gone extinct. These were the Arthropluridae. They were once believed to be their own class, but more recent research has suggested that they're actually a basal and primitive offshoot of the millipedes in the class Diplopoda. Although, like so much of arthropod phylogenetics, there's some debate and some uncertainty about this. The Arthropluridae first emerged about 423 million years ago, about 5 million years after the first millipedes began to appear in the fossil record. One of the most important features linking the Arthropluridae to the millipedes are their fused body segments. Modern millipedes exhibit what are called diplosegments, which is when 2 adjacent segments fuse together, creating a larger dual-part segment with 2 pairs of legs instead of just 1 pair of legs. Their body is thus a chain of these diplosegments, these double links. However, the nature of their respiration is an unresolved mystery. None of the known fossils display any spiracles, which are outer pores for the trachea, the internal tube system that many arthropods use to breathe air. On that note, there's no evidence of lungs. There's also no evidence of gills either. If these were found, it would suggest that the Arthropluridae still lived in an aquatic environment. As we simply don't have any fossil information on their respiration, some scientists assumed that they breathed air and lived on dry land, as that's what all known modern millipedes do. But it goes without saying that this assumption is rather fragile, and could be either supported or disproven with the next discovery of an Arthropluridae fossil that just so happens to have fossilized spiracles, or lungs, or gills. In fact, one such Arthropluridae genus, the E. arthropleura, is quite primitive and basal, and is believed by some to be aquatic, or to at least better represent an aquatic origin of the clade than any other fossil. Environmental data suggests that the Arthropluridae lived in humid coal swamps, where they burrowed through the muck and the muddy soil to find vegetation or decomposing biomass to eat. During the late Devonian and the Carboniferous, vegetation became abundant. Predators were few and far between, and the oxygen levels in the atmosphere were high. This created an environment where the Arthropluridae could grow to enormous sizes. Some of these millipede cousins grew over 2 meters long, making them among the largest arthropods to ever walk upon the face of the earth. This relative golden age, this era of plenty for the myriapods, would soon come to an end. 305 million years ago, the Carboniferous rainforest collapse caused a massive drop in their numbers, and the survivors, the rest of the Arthropluridae, were either outcompeted or killed off by the newly evolved predators in the early Permian, like the newly dominant vertebrate tetrapods. Another extinct millipede offshoot that existed around this time were the Archaepolypoda. Now, these emerged around 430 million years ago, and they existed until the end of the Carboniferous, likely being wiped out when the climate changed and their habitats were destroyed. The Archaepolypoda weren't nearly as large as some of the Arthropluridae. Most of the Archaepolypoda can be measured in inches, like the Archidesmida and the Cowidesmida, which both grew to be about 3 to 4 centimeters long. And then there was the Paleosomatida and the Euphorboreida, which grow to about 6 to 12 inches long. Now you might have noticed that at some point, the myriapods transitioned from an aquatic environment to a terrestrial environment. It appears that this transition happened approximately 450 to 430 million years ago, because before then, in the fossil record, we see aquatic myriapod ancestors and cousin species, and after that time period, we see centipedes and millipedes, which only lived on dry land. The presence of spiracles is usually a dead giveaway. Spiracles are the pores connecting the outside air to the inner tracheal network of the arthropod, so that they can breathe in terrestrial, open-air environments. They don't work underwater, so any arthropod that has spiracles necessarily has to have lived on land. In fact, the oldest land animal, or the first animal species to ever come out of the ocean and colonize dry land, was a myriapod. I should clarify that this is the oldest one that we know of. It's the oldest animal fossil with features that suggest it breathed air. Specifically, it was a millipede-like myriapod called Pneumodesmus pneumani that lived approximately 428 to 414 million years ago, in the later days of the Silurian. So an ancient millipede was the first animal that we know of that adapted away from the marine biome and towards the terrestrial landscape. While it's always a bit tricky to reconstruct the past, especially an evolutionary event that happened hundreds of millions of years ago, there's enough evidence in the fossil record to piece together a rough picture. The first myriapods to colonize dry land would have at first been browsers operating in a coastal or freshwater environment. Here they would have come into proximity of the land and its many bacterial colonies and fledgling ecosystems of young plants and fungi. Long ago, the surface of the world was bare bedrock, but millennia of alteration from the growth of chemolytic fungi and then plants with their rock-splitting roots created the first soils. The early myriapods would have found this terrestrial environment tempting and intriguing. Still near the water, the land was moist and muddy, but coated with strange things like mosses and hornworts and other small, simple vegetables. The myriapods would have colonized this ripe habitat, and for a time they would have enjoyed the ample food and a near total lack of predators. For this brief evolutionary moment in the sun, the myriapods ruled the earth. It's also possible that the ancestral Symphyla and Parapoda, those tiny creatures with their extremely difficult to find fossils, transitioned onto dry land either around this time or even earlier, but we simply don't know for sure. Their fossil record is virtually non-existent this far back in time, but the modern genetic analysis strongly implies their existence here. If this is the case, that the Symphylans and the Parapoda were the first to colonize dry land, but we simply don't have the fossil evidence to support that claim, then we might hypothesize that Pneumodesmus pneumani colonized a terrestrial soil habitat that was already populated with many small myriapods. If this is what happened, then we could hypothesize that those early Symphyla and Parapoda would have massively contributed to these early ecosystems and to the raw carbon biomass in the soil due to the sheer quantity of the waste they produce, their detritus, their exoskeletons, and their dead bodies being mixed into the soil and feeding all the plants and fungi and all kinds of microbes. These early Symphyla and Parapoda would have been ecological vehicles. Compared to the fungi and the plants, They would have been very fast-moving, very efficient movers of nutrients and propagators of ecological cycles. It's possible that these Symphyla and Parapoda, and of course the millipedes, who either came maybe before or more likely after them, also contributed to the development of soil and the propagation of terrestrial ecosystems that made them so inviting for other life forms to come and colonize them. And this itself acted as an engine of biodiversity in the early terrestrial world. And all this activity would have been a great Paleozoic engine of biodiversity. This golden age for the early myriapods went on for about 10 million years. There was about 10 million years of the herbivorous and detrivorous myriapods and other newcomers building up the first terrestrial food webs, and then the first true centipedes arrived and invaded the landscape. These carnivorous myriapods would have been able to thrive in the early terrestrial ecosystems, as they were some of the first land-based predators ever. This was their world now. They usurped the millipedes as the dominant ecological actor, and they evolved and diversified as they expanded throughout the world's soils, pillaging the young terrestrial ecosystems with their ample supply of arthropod herbivores and detrivores. It's not clear if the centipedes evolved their carnivorous diet in the oceans, or if they evolved it after coming onto dry land and adapting to an ecosystem already full of smaller animals. As part of their evolution, the ancestral predatory centipedes developed venom and a means to inject it. The first pair of legs was adapted into a pair of specialist organs called forcipules. The forcipules are unique to centipedes, and they take the form of small pincers that are loaded up with venom glands. For 50 million years, the centipedes used a simple 4-chemical cocktail that they had originally evolved. This was the original recipe, and they used it for about 50 million years. But after this, stuff like horizontal gene transfer from fungi and bacteria began to affect the centipedes and noticeably alter their venom. And after a few million years, you could look at species after species across the Chilopoda clade and see a wide diversity of venoms, each having been adapted to specific prey and specific environmental conditions. But of course, we'll get into this more when we get to our episode on the centipedes. For now, I think we've come to a pretty good stopping point. We explored the early evolution of the myriapods, from their emergence and diversification in the oceans to their colonization of dry land habitats. This is a good place to stop, because at this point, all the major myriapod lineages have emerged and diversified. They've all made it onto dry land, and everything is set up so that we can explore the terrestrial evolution of each individual group in the following episodes. Next up, we're going to do a deep dive into those mysterious, poorly understood, probably very basal and primitive Symphyla and Parapoda. After that, we'll be exploring the millipedes of the class Diplopoda, and then the centipedes of the class Chilopoda. And of course, we'll end the series with an episode on the relationship between humans and myriapods. There's also going to be a surprise episode thrown in here too, so subscribe to the channel and check it out. If you enjoyed this episode, then give it a like, or if you're listening on the Apple Store, give it a 5-star review. If you want to support the podcast in other ways, consider becoming a Patreon supporter or checking out the official store. Whatever you do to support the show, I really appreciate it. And as always, thank you for listening.